Everyone. So it is a pleasure to start this session about the best abstracts in bioinformatics for this meeting. I'm Danilo Augusto. I'm an assistant professor at the University of North Carolina in Charlotte, and I will be moderating this session today. We will start, we will have 10 minutes per presenter, so I hope you keep time, and then you can ask questions, 5 minutes for questions, okay? Our first presenter is Ahmed Mustafa. I'm sorry if I'm mispronouncing your name, but he is an associate professor at U's College of Medicine and Provincial Clinical Lead in Genomics. He also directs the histocompatibility and immunogenetics lab at St. Paul's Hospital. He's certified by American Board of Histocompatibility and Immunogenetics and focuses his research on integrating cutting-edge genomics into clinical practice. So, welcome.
Thank you so much for this nice introduction. And I would like to take this opportunity to thank the Abstract Committee for selecting my talk for an oral presentation. Okay, this is, I don't have anything to disclose. So, as you know, our field started in 1958 when Jean Doucet was working in his lab, and he was mixing some leukocyte with plasma from individuals who have been multiply transfused. And then he found a very interesting phenomena, which is sometimes these cells get agglutinated and sometimes not. But the other thing he noticed is that they don't agglutinate their own self. And then 10 years later, this was the first 8 HLA antigen to be discovered, and it was actually published by a number of publishers, and these are later known by the WHO Nomenclature Committee. And in 1971, I think, it's not moving, it's freezing. So in 1971, the idea of the broad and split came up when they found that there is an anti-sera directed against the A9, does not detect the A9 anymore. And now this was actually the foundation of another new category, which we know as now the cross-reactive group. And by the 1990, this cross-reactive group was very well known, but it did not stay for a very long time until we know that now antibody does not bind to the antigen. Antibody actually bind to a specific part of the antigen known as the epitope. And because of this epitope discovery, this opened another field of what we call now, or what we are known now about the molecular matching. And we have already been talking about this since yesterday. And there is a lot of algorithm present in the market that can predict this HLA matching, starting with the HLA matchmaker by Rene, and now there is a lot of other more algorithm has been developed. But for the purpose of today's presentation, I'm just going to focus on PIRCHE and its two subcomponents, PIRCHE T2 and PIRCHE B, which is the rebranding of PIRCHE 2 and SNOW. And I'm also going to talk about the HLA Eplet Registry. So basically, the HLA Eplet Registry is the updated of the HLA Matchmaker, because if you are still using the HLA Matchmaker, you will notice that the database are not updated regularly, but the HLA Eplet Registry is updated same as the IMGT. So every 6 months, you get a new database that is updated. So what is the HLA Eplet Registry? Basically, if you have a patient who is typed as DR beta 1101, and you are asked to pick between two donors. One is 1301 and one is 1201. Both of them are one antigen mismatch, but when you look at the number of epitope, you will see that donor 1 has only 1 epitope mismatch, and donor 2 has 4 epitope mismatch. So based on this calculation, you will predict that donor 1 will perform better than donor 2. And this is basically the idea of the HLA Eplet Registry. On the other hand, the PIRCHE T2, which is previously known as PIRCHE2. Basically, if you have a mismatched donor HLA, this will be internalized by the patient antigen-presenting cells, and then it will be degraded into small peptides, and then these small peptides will be presented into the patient MHC molecule. But because of the MHC restriction, only a portion of these peptides will be presented. So for example, in this case, you are starting with 5 peptides, and then you will end with 3 peptides. And in that case, this patient or post-transplant will take a score of 3. And PIRCHE actually is a little bit a complicated algorithm. It actually has 2 kinds of filtration. The first filtration is called the snowflake, and then the second filtration is called the snowball. So basically, the snowflake, it actually looks at the solvent accessible surface area, because in order the antibody to bind, you need something that is already exposed on the surface. And then the second stage of PIRCHE-B, it actually looks at the protrusion or the geometry of the HLA antigen, because antibodies prefer concave area. Also, it doesn't prefer flat, so it's not either concave, but it prefers the concave over the convex area. So if you have a donor kidney cell and express HLA, this will be recognized by the patient antigen-presenting cell, and it will become internalized, and then this will be expressed on the patient MHC. This will result in activation of the CD4 positive T cells, and then in turn, this will activate the plasma cell, which secretes antibody, and it can form the donor-specific antibody, which can result into AMR, or it can go to a different pathway like the CD8 positive T cells, and it actually activate and it can result in cell-mediated rejection. So for today's talk, I'm going to focus on these 2 epitopes, or the B cell epitope and the T cell epitope, because as we learned yesterday that we need these 2 linkage between them for a proper immune system. So you cannot rely on one and leave the other. So we started this project by collecting a sample from our cohort patient in Saskatchewan, Canada, 626, but for some of these data were removed because they were missing HLA typing and they were very old. We could not retype them again. So we ended by 594. And this patient has been followed up for developing of donor-specific antibody. So as you can see here, 104 out of the 594 individuals developed donor-specific antibody, but some of these individuals, as you can see here, develop more than 1 donor-specific antibody. So some develop double, some develop triple, and some even develop more. So the total number of events was 146 total events. And in the next stage, we actually divided this cohort into 2 parts. So donor-specific antibody and donor-specific antibody negative. And then we started to look at the Eplet Registry score. And as you can see here, the cohort that has the donor-specific antibody had a higher Eplet score. And this was also consistent when we looked at the PIRCHE T2, and also consistent when we looked also at the PIRCHE-B score. And we have seen yesterday when we were looking at every different algorithm, they all work synergistically at the same time. So this is nothing new that I'm presenting. But this actually raised the idea is what at the level of the locus, because in the previous slide, this was just like we're looking at the whole cohort. But even when we looked at every single locus here, A, B, C, D, R, and DQ, and we couldn't do the DP because some of these patients were old typed and they didn't have the DP typing. So you can see that with every time we have a locus or a donor-specific antibody with any locus, the Eplet registry score was higher in the presence of DSA. When we did this with the PIRCHE, we also find very consistent result. You can see here the A was not significant, but there was still a trend at higher PIRCHE T2 score. And then also when we did the PIRCHE-B or the SNOW, it did not get significant result with the HLA-C or DR, but there was a trend to show that the score was a little bit higher, but it did not reach enough significance. But what we found actually very interesting here is when we combine the locus together, we can get a significant result. So when we put the total score of the class 1 or the total score of class 2, it was all significant in all these 3 algorithms. So we decided, because we were comparing between these 3 algorithms, is to see something more consistent so we can make sure that we are actually comparing apple to apple. And using the ROC curve and the area under the curve, we came up with some cutoffs. So this is the cutoff of every single algorithm that you see here for class 1, and then this is the cutoff for each algorithm for class 2. And then after we calculated the algorithm, we said we can now stratify our patient into high risk, moderate risk, low risk. So high risk, we decided that these are the patients that will have the high score for the 3 algorithms. Low risk will have like below the cutoff for the 3 algorithms. And then moderate risk is if you have 1 or 2 scores high and the third is not high. And then when we followed our cohort, we found actually that this idea is working perfectly fine, because even when we're looking at the donor-specific antibody survival or probability, you can see that the worst outcome was with the high-risk patient who have the 3 algorithms high. The best outcome when the 3 algorithms were low, and then they have a group in the middle which has 1 or 2 of the algorithms were high, or above the cutoff, I mean. And also, because someone may argue maybe this did not follow the same rule with the antibody-mediated rejection, but when we looked at the AMR, we found that actually that the AMR also followed the same trend as the development of this DSA. And we have been asked by a reviewer is, what about the tacrolimus level? Maybe this group of patients was not adherence enough to the tacrolimus level. So we actually compared the tacrolimus trough level between the AMR group and the patient who did not develop AMR, and there was no significant difference between the trough level of tacrolimus. So that shows that mostly the risk is assessed by the algorithm that we are using, not because of the medication. And we also hypothesized that maybe we want to see at the test T-cell mediated rejection, because most of the research coming from this algorithm are focusing on B. But if you look at my first diagram, both arms are working. Both the CD4 can activate the plasma cell, but also can activate the CD8 cells. And what we found here is only PIRCHE T2 has a significant result with the CMR, but neither the Eplet Registry or the PIRCHE B. And we thought that maybe when the PIRCHE 2 is higher, it actually promote more clones or more CD8 to be activated over the plasma cell. We are not sure about the mechanism why PIRCHE T2 has significantly increased with the T-cell mediated rejection. So in summary, molecular mismatch scoring informs alloimmune risk in kidney transplantation. Eplet Registry and PIRCHE predict DSA. PIRCHE T2 uniquely predict T-cell mediated rejection. High-risk group based on algorithm cutoff show the lowest DSA and AMR free survivor. And then lastly, epitope-based algorithms have strong clinical utility for risk stratification and outcome prediction. At the end, I would like to thank everyone who contributed to this research. Hezo, she's the graduate student who did all this work, and our pathology resident, and also Eric Wang, who was here last year, and he was a medical summer research student, and he is the actual founder, or he is the one that started all this project. My collaborator at University of Saskatchewan and University of UBC, and my team at the HLA, and all the funding agents. Thank you so much.
Thank you. We have time for one quick question.
Hi, Ahmed. That was great data. Thank you so much. I have a question about your tacrolimus levels. So was that over a specific period of time before the patient developed DSA? Did you look at a certain window?
Yes.
Because I think it matters.
Yeah, so we actually looked at different windows, and this was a consultation with the nephrologist. So what he advised us is the first 6 months, and then and then 6 months before any biopsy. So we did it in intervals. So we measure the first 6 months, and then we measure at every. But what you have seen here was only one of the measurement we used. But we actually measured the tacrolimus all over the entire period. But we focus more on the first 6 months.
Okay.
Yeah.
Okay, so we're running out of time, but one more. And
Well,
I'll ask
great.
the others to ask a little bit later.
So thanks again for presenting my slides yesterday, where I was actually quite critical on combining mismatch scores because it doesn't really reflect the biology. Why do you combine 3 mismatch scores, 2 of them being B-cell based and basically describing the same biology? It doesn't really make sense to me.
Thanks for this great question. So the idea is the Eplet Registry score is what we use in our lab as a clinical. So we actually have a clinical report in our lab and we put the Eplet Registry score because this is what they use for guiding immunosuppressive. So if it's above a certain cutoff, they increase the trough level. So in order to convince them to move to PIRCHE or to move to another score, we have to do a comparison. So the basic idea here was only to do a comparison between a score that had been established in our lab and to compare it to one of the newer scores, like the PIRCHE score, and to see how both will act. The idea when we found that 3, this was all by chance. We didn't intentionally, but when I was reviewing your slide, yeah, it makes sense that we should not use them as an additive, and I totally appreciate that.
Always take the biology in your rationale. I think that's important. Thanks.
Yeah, thanks.
Thank you very much for the great presentation, and thank you for all the questions.